Hello, everyone, and welcome to today's webinar. This webinar is hosted by Jen and sponsored by Elogen. Today's topic is accelerated protein screening with ENFINIA Linear DNA and GeneFrontier's cell-free PUREfrex. Thanks so much for being here. I'm Uduak Thomas, Senior Editor at Miriam Liebert, a Sage company, and it's my pleasure to be your host for today's webinar. Let's meet today's speakers. Dr. Ebihara Takashi is Chief Operating Officer at GeneFrontier Corporation. Dr. Galit Meshulam Simon is Associate Director for Commercial Applications at Elogen. We'll get to their presentation shortly, but first, some background on today's topic. Transcription Translation Systems, or TXTL systems, are powerful tools for screening recombinant proteins and antibodies, but they frequently involve time-consuming bottlenecks during the synthesis of DNA templates. Although linear double-stranded DNA templates accelerate workflows, many commercial suppliers have struggled to provide the necessary length, complexity, or accuracy required to effectively replace plasmid or PCR-based approaches. There is a need for more efficient and reliable DNA template solutions to fully leverage the advantages of TXTL systems in applications such as protein screening. Elogen's ENFINIA Linear DNA improves TXTL screening by successfully addressing several bottlenecks associated with traditional DNA template synthesis. So in this webinar, our experts will present a case study demonstrating the exceptional properties and high-fidelity performance of ENFINIA Linear DNA as evaluated in GeneFrontier's cell-free protein synthesis PUREfrex system. These results showcase ENFINIA's performance in terms of DNA purity, transcription efficiency, and robust protein expression for various constructs. So what are some key takeaways from this webinar? How this powerful combination eliminates the need for cloning or PCR amplification, which significantly streamlines TXTL workflows. You'll learn how to reduce hands-on time and enable higher throughput protein screening for synthetic biology, protein engineering, and drug discovery. You'll also learn how to apply ENFINIA Linear DNA for challenging sequences such as long repeats, GC-rich promoters, and homopolymers. At any point during today's webinar, if you have a question, you can enter it in the Ask a Question panel on your screen. All right, let's get started.
Hello and welcome to the webinar. My name is Galit Meshulam Simon and I head Commercial Applications Development at Elogen. Joining me today is Takashi Ebihara, or Ebi, the COO of GeneFrontier Corporation. GeneFrontier has been focused since 2010 on advanced cell protein expression technology, particularly the PUREfrex system. Thanks for joining us. The theme of our webinar today is from cell-free DNA synthesis to cell-free protein screening. In the next hour, we will demonstrate how Elogen's ENFINIA Linear DNA, which is synthesized in an entirely cell-free process, accelerates protein expression and screening in GeneFrontier cell-free PUREfrex system. We will start with a short introduction of both companies and their technologies, and we'll continue with data demonstrating that by leveraging both technologies, we can accelerate the path from design to data. Elogen, based in the San Francisco Bay Area, United States, was founded in 2017 by Matt Hill to address the growing need for scalable, versatile gene synthesis. Elogen's cell-free technology overcomes the limitations of conventional gene synthesis platforms that rely on cumbersome cell-based methods. Our production platform is highly scalable and adaptable to meet the growing demand for gene synthesis in synthetic biology, healthcare applications like cell and gene therapy, and mRNA therapeutics and vaccines, and all that without significant investment in resources and infrastructure. Let's talk a bit about conventional gene synthesis. So this is an hierarchical assembly process where short gene fragments are assembled and cloned to correct synthesis errors and to obtain yield. The process starts with short oligos that are typically smaller than 2 kb, those are being stitched or assembled together into larger subunits using methods like Gibson or Golden Gate assembly. This process is repetitive, time-consuming, and error-prone, introducing sequence errors at each step. To isolate the correct sequence, the DNA is cloned into plasmids, transformed into bacteria, then colonies are screened and sequenced until the perfect clone is found. Then a large-scale culture and plasmid prep will be typically needed to obtain sufficient yield. Elogen patented cell-free cloning technology brings a lot of value to our customers by overcoming the limitations of traditional cell-based methods. We synthesize near clonal quality double-stranded linear DNA that is longer and with higher complexity than offered by conventional gene synthesis provider, and it can be shipped within a week. Early in 2023, Elogen launched ENFINIA Linear DNA, providing researchers with double-stranded fragments of up to 7 kb. ENFINIA Linear DNA is NGS verified and ships as fast as 6 to 8 business days. All our linear DNA is produced using the cell-free process and shipped globally from our facilities in the San Francisco Bay Area, United States. The speed, length, and complexity of ENFINIA linear DNA has set the bar for next-generation gene synthesis. Instead of paying more and waiting weeks for long and complex DNA to be cloned in a plasmid, researchers can now get NGS-verified quality DNA without the wait. On the right-hand side, you can see our extremely low error rate of 1 to 70,000 per base in average. This is an estimated accuracy of 99.999%. This error rate is much lower in comparison to other linear DNA providers. Since ENFINIA linear DNA offers near clonal accuracy, high complexity, and length up to 7 kb, it provides a rapid, accurate, cell-free template for all cell-free transcription and translation reactions. I will talk a bit about the main Elogen products, which are all built on ENFINIA platform. So the first one is the one that we talked about until now. This is ENFINIA Linear DNA product, where you can get longer, more complex genes and can do it faster. We realize that many of our customers are using ENFINIA Linear to reduce fragment assembly and to streamline cloning. From this reason, and from the fact that plasmid remain still essential tool in many workflows, we launched in January 2025 ENFINIA Plasmid DNA. So ENFINIA Plasmid provides long, complex sequences in a ready-to-use plasmid format. Here you can get up to 15 kb genes, and the product will be shipped within 10 to 15 business days, still very fast. Our latest product, which we launched in September 2025, is ENFINIA IVT-ready DNA. mRNA is a programmable therapeutic platform with many applications in infectious diseases, vaccines, cancer, and genetic disorder. Despite its rapid adoption, which was highlighted by the COVID-19 vaccines, scalable production still remains limited by issues of speed, fidelity, and batch variability. IVT reactions using T7 RNA polymerase are the standard method for mRNA synthesis, but there are still many challenges with the use of plasmid-derived templates, including poly-A tail instability and bacterial impurity. To address this, Elogen launched IVT-ready DNA, where our cell-free DNA synthesis platform rapidly generates high-quality linear IVT-ready templates with defined poly-A lengths of 70 to 130 base pair. After Elogen introduction, let's hear now Abby presenting GeneFrontier and PUREfrex system.
Thank you for introduction. I'm Abby from GeneFrontier. From me, I'd like to introduce our cell-free solution, which is PUREfrex. Let me start from who we are. We are GeneFrontier, based in Japan, and we are a small biotech, but since 2010, we are focusing on cell-free area. And our mission is to inspire the world with our unique cell-free technology, which is PUREfrex. Okay. And PUREfrex is one of the advanced cell-free technology. And cell-free is basically breaking the cell in advance to break through the limitation of the living cell system. But we are taking one step further, which means we use only necessary molecules for in vitro transcription and translation, or IVDD. So we are taking a breakdown and building up approach. Actually, we are based on a pure system, which was reported by Professor Ueda on Nature Biotech 2001, and then in Tokyo University. And this is a very one of the great groundbreaking technology in synthetic biology field. So it's a rebuilt type cell-free system, I would say. Just adding DNA here, totally molecular-based IVDD system works on like this. And you can get your protein just in a few hours. So I would say it's more like an integrated enzymatic reaction. Okay. And based on that pure system, we have been collaborating with Professor Ueda, the originator of the pure system, so long, and we improved and upgraded the concept of the pure system and developed the PUREfrex as a reagent kit. So I would say it's an advanced version of the pure system. And now, PUREfrex is used not only for the basic research, but also for many, many applications. For example, combination with the cell-free DNA synthesis method, you can establish a high throughput expression platform even without cell alone. And that has good fit to the AI machine learning approach, as you can imagine. And with precisely control of the molecular basis construction, we are proud of the good reproducibility like this. And you can expect the fast and scalable reaction with PUREfrex. Even go to the liter scale, or even you can go to the smaller, like a nanoliter scale as well. And it has good fit to the directed evolutionary screening, based on an in vitro display such as ribosome display, mRNA display, or cDNA display. And you can easily expand your modality to the non-canonical amino acid field. This is an example of the AMBER separation, but it's easy to be done by PUREfrex. And PUREfrex has a robust system, so it is compatible to chemicals such as lipid or nanodisk. And that enables you to tackle with membrane protein or difficult to express protein with unique approach. And with vesicle format, like a ribosome, enables the totally artificial cell, and it's getting hot area in synthetic biology field. So something like this, PUREfrex is expanding its application, and that reminds me of the, biological diversity studied from the once central dogma established. And now you can design the central dogma as you like with PUREfrex, and that expands the huge diversity in the application. And that kind of analogy really inspires me a lot. And I hope I can share that excitement with you. Okay. And from here, I'd like to look into some example with PUREfrex. And this was done by one of our collaborators, Dr. Matsunaga and Professor Tsumoto in Tokyo University. They combined the in vitro DNA synthesis method, in this case, NA builder assembly, and PUREfrex. It's a cell-free combination. And that enables the high throughput and sophisticated validation platform for VHH and its target protein, just 2 days. And it's very high throughput manner, and you can expand the AI machine learning approach together with this kind of platform. And if you'd like to know more in details, it's a paper, so you can check it later. And that was a BHH and target protein, but you can express another modality as well, of course, like a FAB or SCFB, or even difficult complex molecule like a bispecific you can express with PUREfrex. And full-size IgG can be expressed. And here's the example from the third party, Bonham Therapeutics in the US. They are trying to develop the unique immunocytokine, and they expressed a bunch of mutants of their immunocytokine with PUREfrex, and they moved on to the cell-based assay without purification. And that is another advantage of using PUREfrex, because we lowered contaminant, including LPS, as much as possible. Okay. And except antibody-related molecules, you can also express the cytokine or hormone. This was done by Novo Nordisk, as they succeeded in the expression of the functional insulin with PUREfrex, with precise control of the redox state. And you can do that with PUREfrex very precisely. The redox state is very important for the protein, including disulfide bridges. And with that, the research team from Stanford University, they reported this table in their paper. They tried to express 20 different types of therapeutic molecules, which are from the hormone or cytokines, including GLP-1, which is red hot modality nowadays. And they try to express those modalities in different type of cell-based system. So including PUREfrex here. Pure means PUREfrex. And the other three are conventional cell-free system, which means using the lysate. An interesting point is only PUREfrex succeeded in the expression of all those 20 different type of molecules, while the other conventional cell-free system failed in some of them. So I believe this is showing the versatility of the PUREfrex for the expression of those kind of molecules, and which is good as an expression platform. And from the other third party, Roche Diagnostics, for example, they used PUREfrex for the expression of the bunch of FAB mutants for the development of the antibody. And after the expression of their clones with PUREfrex, they moved on to the high-throughput kinetic analysis with BioAccord without purification of their product, which is great. And the other example is from a Japan-based biotech, iBody. They have unique technology for generation of the human antibody called EcoBody. They can generate antibody just in 2 days, starting from the single B-cell cloning. And PUREfrex is used for the expression of their FAB clones in high-throughput mode. And so they can achieve the antibody generation in very, very short period. And the other example is from another biotech from Japan, Epsilon Molecular Engineering. They also have unique platform based on cDNA display and NGS. That is for the screen of BHH or cyclic peptide. And PUREfrex is used for the cDNA display and expression of the BHH clones for the functional screening afterwards. And the large group, Fujifilm, also started using PUREfrex for their screening service for cyclic peptide. They have unique mRNA display technology, and PUREfrex is used in this process. An interesting point is they are using non-canonical amino acid for their cyclic peptide, and PUREfrex is capable for that kind of application. And they also use the PUREfrex for the high-throughput expression of each cyclic peptide for functional screening afterwards, which is also great. And from publication, Stanford research team established a high-throughput validation platform for the peptide and its binding molecules with PUREfrex. And the other research team used PUREfrex for the high-throughput expression of the antimicrobial peptide. And they moved on to the functional screening afterwards without purification of their product. And Roche Pharma used PUREfrex for their ribosome display for the screening of DHH using PUREfrex. And the other research team used PUREfrex for the ribosome display again, that is for the epitope mapping. You can do that kind of things with PUREfrex in high throughput. And looking into the in vitro display, PUREfrex can be used for mRNA display and cDNA display. And in this paper, the research team mentioned that the PUREfrex is the best platform for mRNA display due to the lowest contaminant in the system, so which is great. And this is a research from the Nature, and they reported that cDNA display with PUREfrex, and that is for getting a bunch of folding data of the protein. And the research is aiming for the next generation of AlphaFold, which is exciting research. And we are very, very happy to support this kind of innovative research with our product, PUREfrex. And we are also working on ribosome display, and you can display the scMV, DHH, or cyclic peptide, and even Fab as it is. So we have some IP around here to fully utilize the PUREfrex for ribosome display or in vitro display. So we have licensed to the large pharma, including Suto, in this area. And again, I'd like to mention, and I'd like to emphasize that PUREfrex has good controllability and reproducibility, and that has good fit to the AI machine learning approach. And that is good not only for the development of your product, but also it's useful for the improvement of the platform itself, and that could give you another advantage in your R&D in this field. Okay. And lastly, I'd like to emphasize the key success factor using PUREfrex, that is DNA. And I'd like to introduce 2 points as a key success factor relating to the DNA. The first one is design of the DNA. So PUREfrex is using a ribosome derived from E. coli. So when you think about the design of the DNA template, you should use E. coli-based codon usage, but that is not good enough. Looking at the N-terminus, like 5 amino acid regions after the starting methionine, if you can choose, you should choose AT-rich codon. You don't need to change the amino acid sequence itself, but if you can choose, please choose AT-rich codon. And that is a minor modification, but that could lead to the huge increase in the yield. So this is the example from the heavy chain from the Hercept team, that this is not only the case. You can see this improvement in most protein like this. So GM-CSF is a remarkable example. So just a modification of the N-terminus using AT-rich codon, that leads to a huge increase in the yield. So we accumulated those kind of know-how and information, and we are trying to disclose as much as possible through our website, manual, and publication. So you can check them on our website for free. And next, last but not least, quality of the DNA is very, very important, I'd say. So DNA could be a bottleneck in therapy research. But recently, we found Erigen, and that seems great to overcome those kind of bottleneck in therapy research. But to be fair, we planned this evaluation. That means we asked 3 DNA suppliers, including Erigen, to synthesize some DNAs to see if they are compatible with PUREfrex and if there is any difference in outcome with PUREfrex. Okay. Actually, we ordered those 4 constructs. Basically, the structure is like this. They are double-strand DNA, and they are coding T7 promoter and SD sequence and gene of interest. It's a very simple construct. But the first one is here, is a very, very simple SFGFP. And the other one is just adding the adding the tag. And two of them having the challenging sequence. That is a 10-time repeat of the PPG derived from the collagen. This is very, very challenging sequence, I believe. And due to this difficulty, actually, a supplier B couldn't synthesize those two DNAs. But Elogen and supplier C succeeded in synthesis of those DNAs. With Elogen's DNA, we succeeded in the expression of all those constructs. So you can see the clear band here, and you can see the fluorescence signal like this. And you can see the difference in the expression level depending on the sequence, but that is a fair result because of the challenging sequence we believe. So on the other hand, in comparison amongst three suppliers, looking at construct 3 and 4 here, even though they are the same sequence, Elogen shows the best yield due to the best quality. From here, I would say the better DNA in quality, the better outcome in PUREfrex. And that will be the key success factor for your research too in cell-free system. So please keep it in mind. That is a take-home message today for you. And with that, I'd like to pass it on to Gil. Thank you.
Thanks, Abby, for this fascinating webinar. We hope that we showed you how ENFINIA Linear DNA and GeneFrontier PUREfrex system work synergistically to accelerate design, build, test, learn cycles and shorten the path from DNA design to data. ENFINIA provides high-quality, ready-to-use linear double-stranded DNA that drives efficient transcription and robust protein expression without cloning or PCR. This streamlines transcription and translation workflows, saves times, reduces cost, and basically enables higher throughput protein screening. Together, cell-free DNA and cell-free protein synthesis deliver a seamless, fast, and cost-effective solution for synthetic biology, protein engineering, and many other life sciences applications. Thanks for joining, and please reach out with any questions.
Many thanks to our speakers for those informative and interesting presentations. We're going to begin the Q&A session in just a few moments. As a reminder, if you have a question, you can enter it in the Ask a Question panel on your screen. Before we start the Q&A, one reminder that this webinar will be available to view on demand in the next 24 to 48 hours. So please share the link with your colleagues who might not have been able to tune in today. Welcome to the Q&A portion of today's webinar. Thank you so much for joining us. If you have a question, as a reminder, if you have a question, please go ahead and enter it in the Ask a Question panel that is located on your screen. Galit, Ebi, thank you once again for your presentations. I really appreciate the insights you provided in this webinar. And I'd actually like to go ahead and get started with the first question that I have here for you. And Galit, I think this one's for you. How do you QC your DNA, and what is the practical meaning of the 1 to 70,000 base pair error rate?
Great. Yes, so currently at Elogen, we used a long-read sequencing to QC our linear and our plasmid fragments. We tested or evaluated several sequencing methods, and for us, the long-read based is the best because we can really, with confidence, sequence through the whole long either linear DNA or plasmid. And so we've chosen that. We can go through areas with the repeats or with high homology like ITRs. So we do that. And in general, every gene that we shipped is passing our QC, which means that at least 80% and typically higher than that of the gene synthesized is full length. We estimate the accuracy of our synthesis method to be 99.999%. And combining those means that when we ship DNA, we expect that at least 95% of the molecules that are full-length DNA will have perfect sequence. And the meaning of that is that for applications like Abby was describing with the PUREfrex system, you can just take the DNA and work with it. Most of your molecules will be sequence perfect. You'll get high results, good results as you've seen. If you are using that to clone into plasmid, it means that you typically will have to screen only 1 to 2 colonies because most of them will be sequence perfect. And all that is really streamline the workflow and save time and of course cost.
Excellent. Excellent. Thank you so much for that. Abby, I think this next question is for you. What is the average yield of protein in the PUREfrex?
Yes, thank you for great question. If you are lucky enough, you can reach to 1 milligram per ml. In case of, for example, GFP, you can see the color of GFP. But for the average, I would say after the sequence optimization, you can reach to the roughly 0.5 milligram per ml. So in case of the ACFB or PEP, we reach to the 0.5 milligram per ml. So that could be the average, I believe.
Excellent. Thank you. And I'm actually going to stay with you because with this next question, what is the maximum size of protein that PUREfrex can express?
Yes, thank you for asking again. So from our experience, we succeeded in the expression of more than 100 kilodalton protein. And one of our collaborators succeeded in the expression of 150 kilodalton protein. And it is a multiple that full-size IgG can be expressed with PUREfrex, and in total, IgG could be like a 150 kilodalton. So you can express those kind of large molecules, or you could go much more beyond IgG.
Excellent. Excellent. Galit, this next question is for you. Are there any specific combinations of complexities that are particularly challenging for your technology, for the Elegant technology?
Thanks. We get that question a lot, so happy to answer. So the answer is that not really. Basically, complex sequences like hairpins or long repeats, they are challenging all the methods of DNA synthesis. But we find that our technology handles those pretty well. And as long as you keep your sequence design according to the instructions that are on our website, it should be fine. So our unique advantage, I would say, that we can handle those and we can ship them relatively fast. Overall, Elegant supports 2 tiers of complexity, standard and high. And instead, all the requirements are detailed on our portal at elegantbio.com. But in general, I would say that once you upload your sequence and analyze it on the portal, which is completely free of any commitment, you will see immediately under which category your sequence falls. And what we hear a lot from our clients is that what other vendor will define as high complexity or will reject, Elegant will still do as a standard complexity. And I think it's really nice, according to what Abby was talking about in the webinar, that the design of the DNA template is super important. And if you can go and optimize it to be rich in 80, you can do that all on the portal. You can try different sequences, see what works for you the best, and then submit it.
And just to quickly remind our audience, if you have a question, please feel free to enter it in the Ask the Question panel that is located on your screen. This next question has to do with shipping, Galit, so I'm going to come back to you. How is ENFINIA DNA shipped?
Right. So basically, as I said, each gene that we ship is QC and sequence verified, and we also quantify the yield, and this you'll be find in the manifest, which is sent with every sample. So the DNA is lyophilized and shipped dry in a 96-well format, and then the client could resuspend and normalize or use as much as they need according to their application. And while we make our products in the United States, in the San Francisco Bay Area, we ship globally. We have clients worldwide.
Excellent, excellent. Abby, these next couple of questions I believe are for you. First one, how can I optimize the design of the DNA template of my protein for PUREfrex?
Yeah, thank you for asking. So we are open for any knowledge or information about the design of the DNA from our website, but also we are making the website which is open for anyone can access to the from them and to just type in the amino acid sequence, and we can design the DNA for your protein and get back to you. This kind of consultation is free of charge. So please feel free to access our website. And once you can get the DNA, that could include all the necessary components such as ketone promoter or SD sequence, everything is included. Yeah. And once you can get the DNA sequence, you can ask the DNA synthesis to edit, of course, for sure, in good quality.
Excellent. Thank you. This next question is asking for a bit of guidance. Do you have any advice for the expression of difficult to express protein?
Oh, yes. Thank you for asking. So for difficult to express protein, we have another solution as additives like chaperones or DSPC, which is making for the disulfide wishes. And also you can combine the lipid or chemicals such as detergent, and sometimes as controlling the physical condition, like lowering the incubation temperature to our standard incubation time is 37 or 30 degrees, but you can go down like 25 degrees below temperature. And sometimes that could help for folding the protein, and that could help sometimes for the difficult to express. But if you have any trouble, please get back to us. We are happy to support your research.
All right, I have time for one more question. Abby, this one is for you. What will be the next version of the PUREfrex? Is there any R&D going on at this time?
That's a great question. Thank you for asking. Yes, we are intensively working on the future version of PUREfrex. And especially, we are introducing the AI machine learning approach for the improvement of PUREfrex itself. And internal R&D is actually working going on. And now we are looking at a longer lasting version of the PUREfrex for better yield at the end. So still it's ongoing. So please stay tuned. And I'm hoping to get back to you, the audience, in the near future. Thank you for asking.
Excellent. Excellent. Thank you both so much once again for your presentations and for taking time to answer a few questions for the audience. We really appreciate you.
Thank you.
Thank you so much.
All right. And that is all the time we have for today's webinar. Before we go, a quick reminder that this webinar will be available to view on demand in the next 24 to 48 hours. So you should get a link in your email, and we encourage you to come back and watch this entire presentation all the way through, including the Q&A session. And we also ask you to share the link with your colleagues who might not have been able to tune in today. So once again, a very big thank you to both of our speakers for being here and for such great presentations. I'd also like to thank today's sponsor, Elegant, for sponsoring this webinar. And last but not least, I want to shout out the superb Sage Multimedia Group for all of their support. Thank you to the team. And most of all, thanks to you, our audience, for being here. For everyone at Gen, I'm Uduak Thomas. Thank you and goodbye for now.